Namaste to everyone. My name is Dr. Sonakshi Maiti Das. I did my BSc, MSc, PhD from Calcutta University. Last 6 years, I was working as Neet Zoology Senior Lecturer in several institutes. Today, I am going to demonstrate one topic from neural control and coordination. Now, this chapter deals with several topics which we have to understand and which we need to have crystal clear concept. From this, I choose a topic which is nothing but nerve impulse generation. So here in nerve impulse generation, we have to know first about nerve impulse. What is nerve impulse? So first we will see the topic nerve impulse generation. Nerve impulse generation is nothing but the generation of nerve impulse of an electrical current. So what is nerve impulse? Nerve impulse is nothing but an electrical current which will flow from one neuron to another neuron. Now why it is required? Suppose if there is encounterment of any environmental stimuli, then the nerve impulse will be generated by our sensory neuron and it will be transported to the central nervous system. Now from central nervous system, there will be processing and again generation of impulse, means electrical current, which will come to the muscle and do the necessary actions. So here nerve impulse is nothing but an electrical current which will flow from one neuron to another neuron. But how we can generate a current? By changing or by creating a potential difference. Now how we can create a potential difference? In our system, in human body, in physiological system, we have ions, right? Ions will have some charges. So these charges, if we separate those charges by some membrane, or by any ways, then there is a chance of development of potential difference and there is a chance to develop electrical current. Now let us understand how it generates and how it travels. Okay. So first, for this we need to know about the basic knowledge. Basic knowledge means the structure of neuron, axon, dendron, dendrite, synaptic knob. I hope all you know about that, right? So first, for the base knowledge, let's say this is the axon. The ground substance of the axon is called axoplasm and the membrane is called axolemma. So axolemma is a lipid membrane or bilayer membrane. So it has some lipids and proteins. What are the functions of proteins? Proteins basically helps to transport the ions, right? To transport the substances, molecules. So here, for our understanding, we need 4 types of channel proteins. So we will discuss that. First channel protein which is present in axolemma is sodium potassium active transporter. It is also called pump or pump. Why it is called pump? Because it can act against the concentration gradient. Generally, the ions will flow from high concentration to low concentration. But here, ions will flow by this pump from low to high concentration. Means it will go against the concentration gradient. That's why it is called pump. Now, why it is called active transporter? Active transporter means it needs energy. Energy in the form of ATP. So here, what is the function of that? It will help to transport 3 sodium ion from in to out and 2 potassium ion from out to in. Coming to next one, which is called sodium voltage gated channel. Third is called potassium voltage gated channel. What is the function of that? As the name suggests, voltage gated channel means it will open and close in certain voltage. But remember that it will act towards the concentration gradient, means the ions will be transported from higher to lower concentration, right? But it is not same or similar to the sodium potassium active transporter. It will always help to transport against the concentration gradient. Now what is the opening and closure voltage? Open at minus 60 millivolt and close at plus 35 millivolt. Means when the membrane potential will reach to minus 60 millivolt, this channel will open. When it will reach to plus 35, it will close. Now what is the function of potassium voltage gated channel? Potassium voltage gated channel will open at plus 35 millivolt and it will close at minus 85 millivolt. Coming to another protein which we need to know about, which is called as sodium potassium passive transporter. Means here transporter. Here we know that this active transporter will send the sodium ions towards outside. So in outside, in ECF, sodium concentration will be higher. In exoplasm, potassium concentration will be higher. What this channel will do, it will send the sodium ion from out to in and potassium ion from in to out. Means it will act just opposite to sodium potassium transporter. That's why this channel is also called leaky channels. And it is passive transporter. Means there is no requirement of any energy in the form of ATP, right? Now this four type of channel protein we need to know. Coming to the stages, but we need to know that how the electrical current will be generated, right? So for that we need four types of channel protein. Now let us understand that how the electrical current or the potential difference will be generated, right? So first of all, first stage is called resting stage. Resting stage. Resting stage means when there is no nerve impulse generation. That time will be called as resting stage. Now don't think that in resting stage the membrane potential will be zero. Actually, it will be maintained at minus 70 millivolt and that potential will be called as resting potential. How it will be maintained or generated? We will discuss that. So first, resting membrane potential will be maintained as minus 70 millivolt. How it will be generated? First of all, by three techniques or by three ways. First of all, by sodium potassium pump. How? I told that by the help of sodium potassium pump, 3 sodium will go out with exchange 2 potassium will come in. So we can say that or we can assume that more positive ion will go out with respect to less positive ion which will come in. That can make the membrane negative. Okay. So here by sodium potassium pump. Second one is by some negatively charged proteins. Where it is present? It is permanently present in exoplasm. So this negatively charged proteins which is present in exoplasm, it can contribute the negativity, right? Third one, that permeability of potassium is higher than sodium, by which? By sodium potassium passive transporter. Means it can send more potassium outside with exchange to less sodium which will come in. So if more potassium will go outside, inside there will be more or more negative, less positive. That's how the permeability of potassium is higher than sodium. By these three ways, the membrane negativity will be maintained. And always remember that the negativity we are considering, it is with respect to the exoplasm, not respect to the ECF or extracellular fluid. Now, by these three ways, the resting membrane potential is minus 70 millivolt. Now, what is happening? Now, the next step is very interesting. The next step is depolarization. Okay, so it is also called polarized state, and second stage is called depolarization. Now, when the depolarization will occur, if any environmental stimulus, if it encounters to our body like light, like heat, any environmental stimulus, then there will be a slight change of potential. Means there will be generation of plus 10 millivolt. Okay, so here generation of plus 10 millivolt. It was already minus 70 and plus 10 millivolt generated. So membrane potential will become minus 60. Okay, and this plus 10 millivolt which is generated by the stimulus will be called as threshold potential. What does it call? Threshold potential. Threshold potential. And whenever there is the generation of threshold potential, membrane potential will change from minus 70 to minus 60. Now I told that if it is minus 60 millivolt, then sodium voltage gated channel will open, right? So here whenever then this plus 10 millivolt change is there, and there will be membrane potential which will be minus 60 millivolt, and there will be opening of sodium voltage gated channel. Now outside the sodium concentration is higher with respect to inside, and I told that this voltage gated channel will always act towards the concentration gradient. Means if this channel will open, then immediately there will be rapid influx of sodium ion, right? So here influx of sodium ion. Now membrane potential was negative, right? So sodium ion, if it is coming into the exoplasm, the membrane negativity will be lost and it will become zero first. But it will not stop, because I told that until and unless it was plus 35, it will not stop, right? So membrane negativity will reach to plus 35, and this potential will be called as action potential. That is changing of the potential from negative to positive, and that's why it is called as depolarization. What does it call? It is called depolarization. So let's check it out. Okay, next check it out. So here I told that first stage is resting stage, second stage is depolarization. Depolarization, what will happen? Minus 7 to 70 to it will come to the plus 35 millivolt. Coming to the next stage, which is called repolarization. As I told that in plus or at plus 35 millivolt, which is called action potential, what channel will open? Potassium voltage gated channel will open. So in repolarization, there will be opening of potassium voltage gated channel. As the potassium concentration is higher inside with respect to outside, immediately there will be efflux, means going out of potassium ion. So here potassium will be, so there will be efflux of potassium ions. If there is efflux of potassium ion, so membrane negativity will be regained. So there will be restoration of membrane potential, that is nothing but minus 70 millivolt, right? That's why it is called repolarization. Means again the membrane will get back its own potential or resting potential. But remember that at minus 70 millivolt, the potassium channel or voltage gated ion channel will not close. So when it will close? It will close at minus 85 millivolt, right? So membrane potential will go beyond minus 70 millivolt, and that stage is called as hyperpolarization. Hyperpolarization means minus 70 millivolt membrane potential will be minus 70 to minus 85 millivolt. Now we can understand that eventually by the help of sodium potassium pump, by the help of sodium potassium transporter, and by the help of negatively charged protein, that hyperpolarization will go back to again repolarization, means the membrane potential will be back to minus 70 millivolt. So eventually the process is resting stage, depolarization, repolarization, hyperpolarization and repolarization. So let's check it out with a very interesting graph. Okay, so how it will happen? Now if we see, it is the y-axis, it is the x-axis. In an x-axis, if we take the time in millisecond, actually entire process will take 1 to 5 millisecond. Okay, so here 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. So 5 milliseconds will be required, and this millisecond or this time 5 millisecond will be called as refractory period. What does it call? Refractory period. Because in this time no new signal can be generated. Only this time. Okay, so 1 to 5 millisecond will be there where no neural signal or no extraneural signal will be generated, because already the process is going on, right? So here if we draw, it is minus 80, minus 70, minus 60, then eventually it will be 0 and it will go to the plus 35. In this axis we will take membrane potential in millivolt, right? Now I told that if there is any change from minus 70 to minus 60, then there will be generation of impulse, right? So here if it is from minus 70 to minus 60, means it will come to the threshold potential, then there will be generation of action potential which will reach to the plus 35. So the graph will be like that. This is this one threshold potential. This process from minus 70 to plus 35 we will be called as depolarization. From plus 35 to minus 70 will be called as repolarization, and this one which is called hyperpolarization, right? Because the membrane potential will go beyond the resting potential. So this one is hyperpotential. So hyperpolarization. So this one or this entire process will take only 5 milliseconds, and it is called refractory period. So I hope you understood the process well, and I hope if you understood the process, you can easily crack the questions which will be there in the NEET. If you see the previous year questions also, if you understood the process, definitely you can crack the question from previous year. Okay, thank you so much.